The overall goal of this Fourier-Based diffraction analysis is to characterize the locomotion of C. elegans in real time as the species moves freely in a three-dimensional space.
This method can answer key questions in the field of neurobiology by describing species' responses to different densities of solution or locomotory mutants.
This method serves as a baseline for Fourier analysis of microscopic species. It has the advantage that every microscopic species will have its own unique Fourier spectrum.
Though this method can provide insight into the locomotion of C. elegans, it can also be applied to other microorganisms.
Generally, individuals new to this method will struggle because finding the nematode in the cuvette under the laser beam can be challenging.
We first had the idea for this method in attempting to learn about swimming behavior from the temporal diffraction signal, itself a noisy signal.
To begin, secure a helium neon laser near the back left corner of the optical workbench and connect it to a power source. Place a neutral density filter between the laser and the sample location so that the laser beam travels through the filter before reaching the sample. Using two front surface aluminum steering mirrors, build a periscope by securing the first mirror after the neutral density filter. Then, secure the second mirror about 10 centimeters below the first mirror to give room to steer the laser beam. Insert a cuvette between the mirrors and align the laser beam and cuvette so that the laser beam travels vertically through the cuvette. Next, secure the photodiode directly across from the second mirror so that its sensor faces the mirror.